Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on GATE Xl Biochemistry 2025. So stay tuned. First question, zinc is essential for the function of option A, carboxypeptidase A, option B, chlorophyll A, option C, myoglobin, option D, vitamin B1. And the correct answer is option A, carboxypeptidase A. Zinc acts as cofactor for carboxypeptidase. It means that it is a non-protein component which is the enzyme needs to function properly. So this carboxypeptidase needs zinc to function properly. Zinc helps stabilize the enzyme substrate complex and facilitate the hydrolysis of peptide bonds. The chlorophyll's function relies on magnesium, not zinc. So this option is not correct because its function relies on magnesium, not zinc. Next is myoglobin. The function of myoglobin is to bind and transport oxygen, which depends on iron, not on zinc. So it is not the essential component for myoglobin. Okay. Next is vitamin B1. Vitamin B1, also known as thiamine, which requires phosphorus to its active form. The active form of this vitamin B1 or thiamine is thiamine pyrophosphate, and it does not depend on zinc. So only this carboxypeptidase A requires this zinc, as zinc is the cofactor for this enzyme. So here it is the correct option. Okay. Next, which one of the following molecules captures carbon dioxide in the C4 cycle? Option A, 1,3-bis-phosphoglycerate. Option B, oxaloacetate. Option C, phosphoenol pyruvate. Option D, ribulose 1,5-bisphosphate. And the correct answer is option C, phosphoenol pyruvate. In the C4 pathway, that phosphoenol pyruvate, that is PEP, act as the initial carbon dioxide acceptor, combining carbon dioxide to form oxaloacetate acid, a 4-carbon molecule. So this phosphoenol pyruvate captures carbon dioxide in the C4 cycle. The 1,3-bisphosphoglycerate is a 3-carbon compound involved in the glycolysis and the Kelvin cycles, but not it is the primary acceptor, CA2 acceptor in the C4 pathway. So this 1,3-bisphosphoglycerate is not the primary acceptor C4 pathway, rather it involved in glycolysis and Kelvin cycle. Next is oxaloacetic acid, that is OAA, is formed in the C4 cycle by the carboxylation of phosphoenol pyruvate, that is PEP. It is the product, not the initial acceptor of carbon dioxide. Next, ribulose 1,5-bisphosphate is the initial CA2 acceptor in the C3 pathway, but not the C4 pathway. So for ribulose, it is related to C3 pathway and it is for C4, and this oxaloacetate is formed by carboxylation PEP, and 1,3-bisphosphoglycerate is formed in glycolysis and Kelvin cycle. Okay. Next, which one of the following methods separate biomolecules based on their hydrodynamic volumes? Option A, anion exchange chromatography. Option B, cation exchange chromatography. Option C, size exclusion chromatography. Option D, thin layer chromatography. And the correct answer is option C, that is size exclusion chromatography. Size exclusion chromatography, also known as gel filtration chromatography, uses porous beads uses porous bits to separate molecule based on their size. Larger molecules cannot enter the beds and elute first, while some smaller molecules can enter the beds and take longer to elute. This effectively separates biomolecules based on their hydrodynamic volume, which is a measure of their size and shape in solution. So, the answer is size exclusion chromatography, which is also known as gel filtration chromatography. Now let's discuss about anion exchange chromatography. Anion exchange chromatography is the method that separates molecules based on their charge. Anion exchange resins have positively charged groups that bind negatively charged biomolecules. Now is cation exchange chromatography. It is also a method that is similar to anion exchange chromatography, but with negatively charged resins that bind to positively charged molecules. Now, thin layer chromatography. The thin layer chromatography is a method that separates molecules based on their affinity. Based on an affinity for the stationary phase, that is a solid material coated on the plate, or their polarity. It does not primarily separate based on their hydrodynamic volume. So, the anion exchange and cation exchange chromatography separate the molecule based on their charge. The thin layer chromatography separates molecules based on their affinity for the stationary phase and also their polarity, but does not primarily separate based on their hydrodynamic molecules. Whereas the size exclusion chromatography separate the molecules based on their size and shape, that is their hydrodynamic volume. Next, which one of the following restriction endonuclease is a blunt cutter? Option A, BAM H1. Option B, EcoR1. Option C is hint 3. Option 4 is EcoRV. And the correct answer is option D, EcoRV. This EcoRV is a restriction endonuclease that cuts DNA at the center of its recognition sequence, which results in blunt ends. As it cuts at the center of its recognition sequence, as a result, it results blunt ends. Okay. Next is BAM H1. This enzyme cuts DNA at a staggered site, creating sticky ends. EcoR1, like BAM H1, EcoR1 also creates sticky ends. Next is hint 3. This enzyme also produce sticky ends. So, this BAM H1, EcoR1, hint 3, all three restriction endonuclease creates sticky ends. Sticky. Ends. Okay, whereas the EcoRV produced blunt ends. Okay, these three cause sticky ends. BAM H1, EcoR1, hint 3 produces the sticky end, whereas the EcoRV produces the blunt end. Next question, which one of the following DNA repair system requires DNA glycosylases? Option A, base excision. Option B, direct repair. Option C, mismatch. Option C, nucleotide excision repair system. And the correct answer is option A, base excision repair system. The base excision repair, also known as BR, is a DNA repair mechanism that specifically targets and removes damaged bases from DNA. So this base excision repair targets and removes damaged bases from the DNA. The initial step of base excision repair involves the DNA glycosylase enzyme, which recognizes and removes the damaged base. So basically, the function of the DNA glycosylase is to recognize and removes the DNA damaged base, and it creates an AP site, that is apurinic sites. Whereas the other repair system like mismatch repair or nucleotide accession repair system may involve different enzymes and processes. Whereas the direct repair, as the name suggests, directly repairs and damaged base without removing it. Now, the direct repair involves enzymes like DNA photolase that directly repair specific types of damage such as thymine dimer caused by UV radiation. These enzymes do not use glycosylase. So, if the question come that which DNA repair system require photolase, then the answer will be direct repair system. DNA repair system requires the enzyme DNA photolase. Next is mismatch repair system. Mismatch repair corrects error that occur during DNA replication. It involves identifying and removing mismatch base. As the name, this is mismatch repair, so it's identify and remove the mismatch bases. But the process does not rely on DNA glycosylase. Next is nucleotide accession repairs. Nucleotide accession removes larger damaged section of DNA, including multiple nucleotides. While glycosylases are involved in some form of a nucleotide accession repair, they are not essential for the entire process. The nucleotide accession repair involves other enzymes that recognize the damage and initiate the accession of the damaged nucleotides. So, the base accession repair DNA repair system require the DNA glycosylase which recognize and removes the damaged base, creating apurinic site. Next question. Which one of the following ion channels open to repolarize the neuronal membrane when an action potential is generated? Option A, Ca2 plus channel. Option B, H plus channel. Option C, Na plus channel. Option D is K plus channel. And the correct answer is option D, K plus channel, that is potassium channel. During an action potential, the rapid depolarization phase is caused by the influx of sodium ions through sodium channels. However, for the membrane to return to its resting potential, that is repolarization, potassium channels open, that is K plus channels open. So, for depolarization phase, the sodium channels, that is Na plus channels, should be opened. Okay. Now, when during resting potential or depolarization phase, potassium channels open, allowing the potassium ions to flow out of the cell, effectively bringing the membrane potential back to negative. Ca2 plus channel. Calcium channels are primarily involved in muscle contraction and other cellular process. So, this calcium channel does not open or closed during the neuronal membrane. It is related to muscle contraction. While some calcium channels are involved in specific neuronal function, they are not the main driver of repolarization. So the main function of calcium channel is involved in the muscle contraction and other cellular process. Next is H plus channel, that is hydrogen channel. Hydrogen ion channels are primarily involved in pH regulation and other cellular process, not the repolarization of neurons. So these H plus channels are involved in pH regulation and other cellular process. Now, Na plus channels are responsible for the depolarization phase of action potential, not repolarization. So, these Na plus channels are involved in repolarization, K plus of neuron, and this K plus is involved in depolarization. Okay. and Ca2 plus channels are primarily involved in muscle contraction. This is the Ca2, and the H plus channels are involved in pH regulation. Next question. Which one of the following is the most sensitive immunoassay? Option A, immunoelectrophoresis. Option B, immunofluorescence. Option C is radial immunodiffusion. Option D is radial immunoassay. And the correct answer is option D, that is radioimmunoassay. Radioimmunoassays, that is RIA, uses radioactive levels to detect antigens or antibodies, allowing for extremely high sensitivity in detecting even very small amounts of substance. Immunoelectrophoresis uses an electric current to separate proteins in a sample. Then antibodies are added to identify specific proteins. While useful for visualizing multiple proteins, it is not as sensitive as RIA, that is radioimmunoassay. Next is immunofluorescence. It uses fluorescently labeled antibodies to visualize the presence of specific antigens under the microscope. It is more sensitive than immunoelectrophoresis but less sensitive than RIA, that is radioimmunoassay. Radial immunodiffusion. Option, that is option C. Radial immunodiffusion involves diffusion of antigens and antibodies in a gel to form visible precipitation lines. It is relatively less sensitive compared to other options. So, on the basis on sensitivity, it will be like that. RIA, most highest sensitivity is RIA. Then is immunoelectrophoresis. So it will take much in line. So I will thus option D, that is RIA, followed by immunofluorescence, that is option B, followed by immunoelectrophoresis, followed by C. So RIA, that is radioimmunoassay, is more sensitive, followed by B, that is immunofluorescence, followed by A, that is immunoelectrophoresis, followed by C, that is radial immunodiffusion. So this radial immunoassay, that is RIA, is most sensitive than any other options. Okay. Next question. Which of the following are peptide hormones? Option A, calcitonin. Option B, glucagon. Option C, serotonin. Option D is thyroxine. And the correct answer is option A, that is calcitonin, and or option B, glucagon. Both calcitonin and glucagon are peptide hormone. Calcitonin is a peptide hormone produced by thyroid gland that helps regulate calcium levels in the blood. Glucagon, also a peptide hormone which is produced by the pancreas, and it raises blood glucose level. Serotonin plays important roles in the body. It is classified as a monoamine neurotransmitter, not a peptide hormone. Thyroxine is an amino acid derivative hormone, not a peptide hormone. It is derived from the amino acid tyrosine. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.